So, thank you, everyone. I'm going to mention a few words about why we are here to celebrate the work and life of Meyer Birnboim, who was professor first of mechanical engineering at Rensselaer for over 30 years. And his work focused on using optical techniques to measure the rheological and optical properties of materials. And in fact, he did a bachelor's in physics in University of British Columbia. He did a master's in applied physics, University of British Columbia, and a PhD in physics at the University of Wisconsin in Madison. The Professor Meyer Birnboim Lecture Series is a joint event, which is really very interesting, between chemical and biological engineering and physics, applied physics, and astronomy. The event alternates yearly between the two departments and the two schools, thanks to the efforts of our deans, Shekhar Garde, Dean of Engineering, and Kurt Brenneman, Dean of Science, who sadly passed away yesterday. So we will miss him a lot. The series topics also alternate between rheology on the engineering side and optics on the physics side. Thus, the series serves as an interface of science and engineering, trying to promote collaboration between scientists and engineers. I am passionate about multidisciplinarity, and I like to mention a quote by Russell Lincoln Acuff, who used to say that nature is not organized in the same way universities are. So we have to collaborate and try to do multidisciplinary research in order to solve the more complex problems. I want to especially thank the Birnboim family for this wonderful idea to start the lecture series and for the generous financial support. Thank you, Joseph, thank you, Rachel, and thank you, Alma. Joseph and Rachel are son and daughter of Meyer Birnboim, and Alma, his wife. Thank you very much. So now I will introduce our speaker today, Professor Julie Biteen. She was born in Montreal, not far from RPI. She majored in chemistry at Princeton University. Then she moved to Caltech for her master's in applied physics. So, you know, physics is there. And she did her PhD at Caltech under the supervision of Harry Atwater and Nathan Lewis, working on plasmonics and quantum dot optoelectronics. Physics again. She carried out her postdoc at Stanford University, where she worked on super-resolution imaging with William Murner, who got the Nobel Prize, shared the Nobel Prize in 2014, Chemistry Nobel Prize, with Betzig and Hill for the development of super-resolved fluorescence microscopy. She is now at the University of Michigan, Ann Arbor, where she is Janine Maddock Collegiate Professor of Chemistry and Biophysics. She's senior editor of the Journal of Physical Chemistry. And I have to say that she has many honors, and I will mention just a few. In 2009, she is of Burroughs Wellcome Fund Career Award. She got 2013 NSF Career Award, 2014 Moore Foundation Sociologue Fellow, 2016 Journal of the Physical Chemistry Lectureship Award, 2017 Biophysical Society Margaret Oakley Dayhoff Award, in 2020 the NSF Award for Special Creativity. So let's just give a warm welcome to Professor Biteen, who is going to talk about what can single particle plasmonics and super resolution microscopy tell us about the living world.
Well, thank you so much. Is this microphone working? Yes? Okay. Thank you so much for that nice introduction, and thank you so much to the Birnboim family for making this visit possible. I've been having a terrific day at RPI. There's a lot of weird connections. One thing is that my father actually did a master's in applied math at RPI, and so it's so cool to see this, this place of learning. But then I also was reading about Meyer Birnboim, and I realized my grandmother is also a Polish Jew who, she's 97 years old, and she moved here, and she's about the same age, and we grew up in Canada. And I see some parallels of this sort of life, life not led by my family that we're getting to interweave into, and so that felt very special. Professor Birnboim had a very nice, as Umberto mentioned, multidisciplinary research program. And I feel like this is also at the heart of my program. And so I'm happy to be here today to tell you some of the physics that we're working on. But you'll see, there's interlaces of a few different themes. So let me tell you about my lab at the University of Michigan. I've been there about 15 years. And at the heart of our program is the idea that we could detect single molecules in a fluorescence microscope. This is a pretty standard microscope, but because of the sensitivity of fluorescence, we can see just one molecule at a time. We can ask, where is that molecule? We can ask, how bright is that molecule? We can sense the local environment, and then we can build up maps of how things are positioned and moving on the nanometer scale in real time, even in living cells. And so I don't have a lot of raw data in my experiment, in my slides, and so I wanted to just start by showing you the type of work that my students are doing. And so what my students are doing is looking on a camera and detecting these molecules. What you see here is this sort of pixelation noise, and then you see that big white spot at the center. And so this is saying that we're right at the edge of the sensitivity limit, but because we know things like that this shape looks different from this pixelation noise, we can identify those molecules pretty easily, actually. My lab also thinks a lot about algorithms for processing the data. And so all the grayscale things here are raw data, but the red line, that's a track where we're capturing in every frame the molecule and watching how it moves. And this happens to be an example of unidirectional motion. So you kind of see it tracing out a straight line. So let me tell you a bit more about the technology, but I'll spend most of the talk talking about the applications. And so what we're doing in fluorescence microscopy, the students have probably all seen that when we do fluorescence, we excite at one wavelength, we're collecting emitted light at another wavelength. And so we do that using a laser. We excite our sample with green light. We use a standard optical microscope, much like the one you'd see in Professor Wirtz's lab if you're up in the physics building. And then we look at the emission, and in particular, we're looking at the redder emission, so that gives us the high sensitivity of this technique. I love it because these are experiments that we can get up and running within about a week when we have new samples, and we can get quantitative information out very quickly. So what we do with this data is a few things. One thing we can do is just say, how bright is that molecule? What's going on? Then the other question we might ask is, where is that molecule? We all know that light has a wavelength. That wavelength's about half a micron. So if you use a normal microscope, your resolution is limited to that micron scale. But what we can do is say, well, if I know this emission is all coming from just one molecule, then I know that that image is the focus spot from my emission. It's an area function if you want to be mathy. And we can fit that image, and we can find with very high precision what the position of the actual molecule is. So we've gone now from the scale of light, which is more like microns, to the scale of molecules, more like nanometers. And what really blew me away when I sort of entered this field, I went up to Stanford to do my postdoc, and I read this paper. And this paper was something I'd never seen before, which is biology. I had taken a very bare minimum of biology before that. And so what I found was, here I told you this is one molecule. This is a Cy5 dimolecule immobilized on a glass slip. Well, what Soyan had done was she took a bacteria cell that you see right here, and there's one molecule, it's a fluorescent protein inside of it, and it played the exact same trick. So the point is that most things in biology are not fluorescent, and so now if we introduce a fluorescent molecule, we could still track it in real time and even in living cells. And this is just a snapshot, but if I play the image, you see that molecule doing a zigzag back and forth, left to right across the cell. So these are the kinds of technologies that my lab is really excited about, and we pursue them in several different directions. One thing is thinking about what do I do with all this data that I'm creating. Another piece of our lab that I won't really have time to go into much today is back to that idea that we could see molecules inside living cells. And so we think a lot about bacteria cells. We think a lot about microbes in general, a little bit about yeast cells. And in all those cells, we can watch how molecules move. We can ask those questions that, sorry to say, kind of bored me when I learned biochemistry about how do proteins interact. I find them amazing when we can actually watch them interacting inside the living cell. And actually, to speak to rheology, one thing that we could do is track sensor particles inside these cells. And again, even though the cells are only microns in size, we can get nanometer scale information about viscosity and accessibility inside those cells. But what I'd like to focus on today is really another half of my lab, and that's where we think about light matter interactions and about using this single molecule technology as a really sensitive probe of the local environment. Just to briefly introduce Plasmonics, what we're thinking about is the fact that, of course, metals have lots of electrons. We know they conduct electricity. When we confine the electrons to a small volume, the charges are going to oscillate. And so we go from having a surface wave in bulk physics to having these local particle plasmons on the nanoscale. And so during my PhD, I thought a lot about the fact that if we take this small nanoparticle and we excite it with visible light that's resonant, the electrons, the charges are going to oscillate back and forth from end to end. And because this is only maybe 100 nanometers big, that's a really large separation of charge over a very small space. And so bottom line, this is a powerful antenna, and it's a powerful antenna that we can interact with visible light. And so during my PhD, I was thinking a lot about, or the field was thinking a lot about plasmonics as ways to enhance optical properties. This is some work that I was doing, thinking about using silicon quantum dots and coupling it to gold nanoparticles and looking at brighter emission because we have that antenna. So the nanoparticles can, or the quantum dots can interact more strongly with the light than they would otherwise. Other people in the field were looking at enhanced fluorescence, at enhancing solar energy absorption, at enhancing spectroscopy, and at enhancing sensing in that way. And so there was a lot of work going on. One might say, what's needed here? What can we say? But the issue that I always had with these experiments were that they were always averaged over large areas of space, over large samples, and there was a huge distribution. And so I came to these projects as a PI thinking, what can single molecule imaging tell us about particle plasmonics? And so the approach that we have, as I said before, is we can look at fluorescence from just one molecule. And so the approach that we have is to put a single dimolecule close by to that plasmonic particle. And by detecting those interactions one molecule at a time, we can really think about the single particle effects and even geometric effects within that space. And so the approach that we're using is this single molecule detection. And one thing that I haven't told you is that there is one challenge with single molecule imaging. If I have a molecule, my lasers are aligned, that stuff's easy, I could see the emission. If I have too many molecules, they're just all going to be one on top of the other. And so what we need to do is have a very sparse amount of dyes in time. And so what we do in my lab is we take the microscope objective, or the microscope cover slip that we put right on top of the microscope objective. This is where I would put any kind of sample on top of this glass slide. And I put a very low concentration of dyes, picomolar, sometimes nanomolar, on top of the slide. And much like if you took a picture of me in low light and I waved my hand and it makes a bit of a ghost, much like that, the molecules that are freely diffusing up in the solution, they just blur out entirely. Their photons are smushed out over many, many pixels. But on the other hand, when the dyes absorb to the slide, I can actually immobilize them long enough to see them. And this is just stochastic. My lab is in a chemistry department, but we're not very good at things like surface chemistry. This is just stochastic. We pick the right pH of our solution so that the dyes are, sticking down to the glass slide. So my images look something like this. This is a movie shown in real time. Every 40 milliseconds, I'm integrating a frame. And what you can see is splashes of those white dots. And every white dot I know is a single molecule. I know it's a single molecule for a few reasons. One is they're all pretty consistent in brightness. And the other is that the width is about half the wavelength of light, which is my diffraction limit that I expected it to be. So these transiently absorbed dye molecules give me bright spots on the camera. And for every bright spot that I see, I could say, where is it? Remember, I can sort of fit it or draw that crosshairs, find the center position, say, how bright is it? I can ask all kinds of questions. And this is a very high throughput experiment. You get many, many, many molecule absorption events over time. So when we start to probe plasmonic interactions, the experiment that we do is to say, okay, here is my gold nanorod. I showed you before a gold nanoparticle. This one is a rod shape, and the reason to make it rod shaped is it gets a little redder, and that resonant overlap happens with my dyes that are also red. And now again, right, my dyes can be free in solution, I won't see them again. They could touch down away from the nanorod, and my hypothesis is they'll be dim, and they could touch down close to the nanorod, and my hypothesis is they'll be bright. So this is work that we started in my lab about 14 years ago. I want to recognize Professor Esther Wertz, who's going to blush in the front of the audience. Esther was the postdoc in my lab who initiated much of this work, and you'll see her name decorated throughout many of these original publications. I'll tell you a bit more about Esther's specific contribution later. But let me start from what Jessica, my first grad student, was doing. And Jessica was looking at a fluorescent protein and asking, can we enhance it with a metal nanoparticle? Now remember, much of my lab is doing bioimaging, and I'll get to the biology applications a bit at the end. But the original inspiration here is, okay, well, we know we can enhance dye molecules. We know we can enhance quantum dots. What about these fluorescent proteins that are our workhorse in bioimaging? And so Jessica looked at this M-cherry. It's just a red fluorescent protein. And what you see is the brightness of the emission has a certain mean value. And when we start coupling it to gold, the brightness increases. So you see that the average increases. It's a linear effect. It's not a blowing us out of the water kind of effect. But the other interesting thing you see is that the width is increasing, the breadth, or the broadness of that peak. And what's happening here is that a molecule that's close to the gold is very enhanced. A molecule a little further from the gold is less enhanced, and we start getting at a distribution. And we've modeled this distribution over time with finite difference time domain simulations. Bing Fu continued this project, and what she did was think about, okay, what is that spatial dependence? And so we have our gold nanorod. She covered it with different types of polymer layers or with sputtered silica, and she established that a dye that's too close to the gold is going to be quenched. We've seen this before across the field, and that's a charge transfer event that's happening. A dye that's around that sweet spot between 5 and 15 nanometers is going to be most enhanced. And a dye out to about 50 nanometers away from the gold surface still feels some enhancement effect until that dies right off. So it's fun in my lab, right? Because there's people working on different projects, and sometimes they talk to each other. And so I told you Jessica was my first grad student. Well, my other first grad student was Beth. And Beth was doing bioimaging side of the lab. And I'm not going to go into the details except to say that Beth was looking at a bacteria cell. And bacteria cells have gram-negative bacteria cells have an inner and an outer membrane. And because of Beth's specific project, she was thinking about this protein that sat on the inner membrane that was labeled with a fluorescent protein. Remember that M cherry? The fluorescent protein was sitting in this periplasmic space, so right there at the edge of the cell. And I loved this experiment because Bing was taking a gold nanoparticle and putting a dielectric over it and looking at the dye. Well, the question is, can we do the same thing with a membrane separating our dye from our particle? And so Jessica started this work, and then Stephen picked it up. And what they did was they put, this is just a cartoon, not data, but they put a cell with the fluorescent protein on top of an array of gold nanotriangles. These are made by nanospheric lithography. And what they found is that there is an enhancement about fourfold of the emission from this molecule that's inside the cell. And importantly, gold is non-toxic to the cell. Nothing is going inside the cell, and we could still see some enhancement. They also showed some resonant effects. So a green dye could be enhanced, but the bluer dye could not. So, we started off these projects, and we said, okay, if we're trying to do single molecule detection, and we want to see our molecules, and we want to have high precision, we want to get lots of photons out of it, and if we can enhance the brightness, we're going to have better probes. And there's chemists working on better probes, but I'm not an organic chemist, so this is a nice way to make a brighter probe. So this is when Esther came into the lab, and we had these great conversations because we said, okay, we're going to enhance the fluorescence, and moreover, we're going to measure the electric field distribution, right? We do all these Maxwell's equation simulations, and we can map out an electric field near a plasmonic particle, but wouldn't it be cool if we could say, oh, well, the molecule here is really bright, the molecule here is less bright, the molecule here is less bright, and really map out that field in real time. We hit a snag pretty quickly as soon as we kind of started thinking it through. There were papers in the field actually trying to claim that they had done this. But basically what we came to, and this is work done in my lab by Esther as well as another student, Ben Isaacoff. And what we came to at the end was we said, if I have a dye that's close to a gold nanoparticle, the center of mass of this coupled system that I'm trying to image is actually basically on top of the gold nanoparticle. The gold nanoparticle has most of the dominance in this system. And so you can see this simulation that Ben did. This is a dipole. It should be emitting symmetrically. And instead, it's not awesome on, it's not even great on my screen. But instead of seeing it being symmetrical, what you see is there's more emission or more electromagnetic intensity on the left. And you can Is that better if I move it a bit closer to my mouth? Okay. All right. Let me know if you can hear me in the back, because I can also pick up the hand microphone, because I have two hands. Okay. So this is a problem, but the corollary is really exciting. The corollary says that the single molecule fluorescence is a really nice probe of the local environment. It's not just, is it brighter, is it dimmer? But if we think about the shape of the emission pattern, we can actually get information out about our system. And so the experiment that Esther did really started our thinking here. And so what we did here is that paint experiment. These are samples of gold nanodiscs that are made at the electron beam nanofab. And what you see is a lot of flashing. And so this is that same paint experiment. Dyes are touching down on the surface. It just increases the throughput of our experiment. And so again, our hypothesis was, okay, a dye that's close to the nanorod or nanoparticle should be bright. A dye further away should be dim. And a dye that touches down on top of the gold should be quenched, because we know there's a charge transfer event that happens. This is not what we saw. And instead, we saw a pattern like this. This is a picture where dyes are touching down completely randomly. The surface coverage should be completely homogeneous. And rather what we see is there's still some molecules out here, and they're dim. Blue is dim here. But the brightest molecules appear to be right on top of the gold. And so what is happening here, and what we later confirmed with some triangle nanoparticle experiments that gave us better control, is that the dye that touches down close to the gold is brighter, it's red, but also its emission appears to be coming from the center of the gold particle, because that coupled system, that dye nanoparticle coupled system, actually its center of mass is at the center of the gold. It's a little more complicated than center of mass, but I find that a really good analogy. So the emission position that we see when we do this single molecule fitting is no longer the actual position of the dye, and rather we've reshaped the emission pattern using a pretty simple geometry, right? Just a gold circle. So Bing went on to look at this in 3 dimensions. Another nice thing of working in a multidisciplinary lab is there were people working with DNA, and the people doing more solid-state physics weren't afraid of those experiments. And so Bing took a gold nanoparticle. Now it's a symmetric particle. She functionalized it with double-stranded DNA, which is pretty rigid on the scale of 20 to 50 nanometers, and then put a dye at the end there. Using some sort of single molecule approach called de-storm, what we did was looked at the emission. And again, if you saw fluorescence from the position of the molecule, what you'd expect to see is a ring that's much bigger than the gold nanoparticle size. And rather, when Bing looked through this, and she has some 3D sectioning too, she saw that the emission moved from the red, which is the actual place, to the black, which is the surface of the nanoparticle. And so we see the molecule fluorescence emission pattern shifting, and it's shifting in a way that we could control using these nanoparticles. Around that time, my collaborator, Dr. David Masiello at University of Washington, wrote this really nice paper, or his student Harrison Goldwyn wrote this really great paper. And what was really gratifying to us is that this intuition that we had been building up over time really was borne out in the theory. And the theory here is very simple. It's really just modeling the dye as a dipole oscillator, and then modeling the nanorod as a dipole oscillator, and looking at the coupling between them, look at interference, constructive as well as destructive interference. And Harrison was able to see the same thing, which is, yeah, the emission pattern should be changed in this programmable, controllable way by the gold particle. So we find that plasmonic nanoantennas reshape the emission from single molecules. And I was talking to some faculty today about the fact that labs kind of go through these cycles. And I'd say we're on to cycle 2. And so let me finish up this overview by talking about cycle 2 in my lab. And now we're finishing up cycle 3 in my lab is the way it goes. Because grad students, y'all are around here about 5 years, and I've been a professor for 15. So, again, Esther saw that the dyes that were meant to be out here appeared to be shifted towards the gold. They are not physically shifted. That's another hypothesis that we tested. Really, the emission pattern is weighted towards the position of the gold because the emission is coming from a coupled system. So we said, well, if we can reshape the spatial shape, what other parameters are out there? And so Stephen went ahead and took our setup. And again, our lab setup is not that complicated, right? A laser, and I should have mentioned this, these are all CW lasers. They're all low power lasers because we just want to see the emission from one molecule. Our laser is coming in to our microscope. We have our sample, and we look at the emission with some careful filtering of the laser light, and we see the patterns on that camera, that pixelated detector. So Stephen introduced, well, it's not really one optic, one box there, and that was a spectrometer. And so what Stephen said was, oh, well, if I put a spectrometer in here, every time my molecule adheres to the surface, actually what I should see is a full spectrum. And so he looked at these molecules. These are some fairly blue molecules, and they're coupled to nanorods that are a little bit more red. you see our signal to noise here, and I think it's an important fundamental piece of these experiments and the reason that we do everything 100,000 times. So what Stephen found is that these blue dye molecules that were close to nanorods, the emission from the coupled system actually got redshifted, and he found that the redder dye molecules got blue shifted. So we're reshaping the emission pattern both in space but also in spectrum, in energy space. So then Curly came into the lab and he said, I'm going to do something even more simple. He didn't, the math was more complicated, but the experiment was more simple. And let's go back to this simple experiment and introduce a single optic right here, and that's a polarizing beam displacer. And so what does the polarizing beam displacer do? Well, the emission that's coming out, it's going to separate S and P polarized light into two channels. And so whereas previously we had a movie where we saw single molecules touching down, now the molecules are going to register in both channels, and by correlating the images, we could start to get a measure of the angle of the emission, which should coincide with the angle of the dye that's adhering on the surface. And so if the emission is similar in the center and off-center channel, well, that's a dye that's at about 45 degrees from the polarizer. And of course, we could put it all on one channel or all on the other. And so Curly did the same experiment, which is, can we couple the dye to the metal nanoparticle? But this time what he asked is, how is that going to affect the emission polarization? And so he started with a dye. It's our favorite dye. It's Psi 5.5. It's just kind of low quantum yield, really wants to be enhanced, really wants to be coupled. And so he took this Psi 5.5 dye, and he measured the polarization angles. And you see that they should be flat, but due to some sensitivity issues, they're symmetric around 45 degrees. So you see a peak around 45 degrees, which means equal chance of being polarized that way or that way. And that makes sense. This is a totally chemistry-free experiment. The dyes are just randomly touching down on the surface. So he then coupled the dyes to the gold nanorod. The gold nanorod overlaps spectrally with the emission wavelength of Psi 5.5. And what Curly found was that whereas previously the molecules seemed to emit all about 45 degrees, now if I have a nanorod that's about 70 degrees, I see emission that seems to be 70 degrees. And if my nanorod is rotated the other way, I see emission looking the other way. And so Curly did this for many different nanorods, each oriented at a different angle. And again, what he sees is that the dye emission seems to be shaped not just in space and in spectrum, like I've shown you already, but also the polarization of the emission is shaped. And so again, using this very simple geometry, which is just a gold nanorod that we buy commercially, we can change the shape of the emission. We can change a lot about the emission properties of the dye molecule. So sort of as some intermediate conclusions, and then I'll take 10 minutes to tell you about the latest work in our lab. What we've found over the years is that a dye molecule near a nanorod will definitely have enhanced, plasmon enhanced, and plasmon changed properties. We can use that to change the brightness, which was our original goal, but also we could see changes in the emission pattern and the emission properties. So let me take the last 10, 15 minutes and tell you about what's going on currently in our lab. And this, of course, starts during the pandemic. And so Saj was a terrific applied physics student who started in my lab, a year before COVID. And so while our lab was shut down, she was just really ramping up on her research. And sorry, not a year before, 6 months before. All right, she was just ramping up on her research, and I said, go read all the papers, right? So the first thing that you do is you read the lab's papers, and the next thing that you do is you find your good friends and what's the review article that they wrote. And she identified about 10 labs at the time that were really working hard in the area of single molecule plasmonics, and she wrote this great review that I thought was nice for somebody who was holed up in their dorm room when we couldn't be in lab. But that really inspired us to think about what are the new directions that haven't been explored. And, in particular, because a large part of my program is doing live cell biology, it would be really nice to make sure that we're leveraging that capability in our lab. And so, one really important theme that emerged was chirality. And, the question is, can we measure chiral signatures in vitro as well as in biology? And can we sense these chiral superstructures? And so there's two reasons this is great. Reason number one is chirality is ubiquitous. So, if I think about this bacteria cell, the flagellum that helps it move is actually chiral. But also we know that bacteria cells make biofilms, and the fibers that come out are also chiral. And obviously the DNA inside the cell is also chiral. So chirality is definitely interesting. But the other reason that I think chirality is an interesting target for us is that these are pretty subtle signatures. The scale of the chirality here, the scale of the differential absorbance or interaction of light with a molecule is very, very subtle compared to the wavelength of light. And the signatures are very small. So, one of our big goals right now is to say, can we use metal nanoparticle plasmonics to enhance those chiral signals? And can we use single molecule detection to measure chirality? So Dan Faust, who's a postdoc in my lab, has started thinking about these projects. And in particular, what you see here, this is an E. coli bacteria cell. This is 2 E. coli bacteria cells. And these E. coli cells, when they go into a certain phase, start building a biofilm. And we all know about biofilms. That's why we brush our teeth, right? They protect our skin. But also, they lead to infections in hospitals and things like that. And so what Dan has been doing is decorating these fibers with molecules that intercalate in there. And these are very preliminary results showing that he could super resolve these fibers. When you look at it on the single molecule scale, he's decorating them each with a molecule. And importantly, these dyes are intercalating in the fiber such that their orientation reflects something real in the biology. And so, one of Dan's goals right now is to think about how does this chirality evolve over the course of biofilm formation. Another thing that we did, and another great tie to RPI, is that Professor Ji-Yoon Kim was working in the lab of Nick Kotov, and Nick said, oh, I also like chiral materials. And that really pushed us to develop our microscope, not just to see polarization, but also to see circular polarization, right, and to sense chirality. And so these are some of the particles that Ji-Young was making that using light in the synthesis could be either right-handed or left-handed helices. And so from her bulk scale measurements, what she saw was there was some circular dichrosum. If I have a collection of these helicoids, they have a different circular dichrosum than the collection of the red helicoids. And so, the question, it's not exactly the question Nick asked me, but it's the question I thought I could answer, is how consistent and homogeneous are these particles across the sample? Just because the net effect is that dominated by one, or is that an average property? And so Zek went ahead and took our microscope, again, back to our basic microscope, and what he did was he introduced some wave plates and a liquid crystal variable retarder in order to shape, now on the excitation side, the light that's going into the sample. The LCVR is necessary because the dichroic in the microscope is actually going to distort the polarization. And so what we do is we take our laser light, which is linearly polarized. We use these wave plates to get it roughly circularly, or to get it circularly polarized. And then we use the LCVR to make sure that the light is perfectly circularly polarized at the position of the sample. And so what you might imagine is a particle that interacts more strongly with the right-hand circular polarized light versus the left-hand is itself right-hand circular polarized. And so this is what Zek saw when he used LCP excitation. This type of particle was pretty dim, but it was pretty bright in the RCP excitation and vice versa. And importantly, as he characterized many, many particles, what he found was most of the nanohelicoids in each ensemble were one polarization that they were supposed to be, but he also saw that there is a distribution. And these are the types of things that once you could get this single particle sensing, you can actually start to go back and inform the fabrication process. So, coming back to, now that we had the chiral microscope, coming back to our biological systems, there are ways to use biology to template patterning of plasmonic materials. And Luis Liz-Marzan had written this paper using some polymers, some biological polymers, to template these long nanorods into a chiral geometry. The nanorods themselves are not chiral, but you put one this way, and one this way, and one this way, and one this way, and at some point you end up with the helix. So what we did in collaboration with Dr. Olesiak Bańska in Poland was we said, what does this do with some more simple materials that she had her hands on, including gold nanobipyramids that are very different scale from these because they could actually intercalate into the biological fibers, and these biological fibers are amyloids. And so what they had observed, what Meche had observed, is that even these very small nanorods that are not being organized into that helix because they're just too far apart for that and too small, they were still showing signatures of the chirality of their local environment. And that made me really excited because the underlying hypothesis here is that the emission is going to be shaped by the local environment. And so Meche actually came to visit for a few months and worked with Saj and Zek, and said, what's the underlying cause of the high circular dichrosum? And Saj did a lot of experiments in this setup with that wave plates and LCVR. So again, can we look at these assemblies, and not just on that mole scale, but on the local scale, can we see a local response? Is it that the small nanorod or the small bipyramid is feeling that chiral environment to give us a signature, or is it a longer ranged order? We have access to the short ranged order. And so, here's her microscope again. And so again, linear polarizer, and then an LCVR that controls the circular polarization right at the sample. And so what Saj did was she looked at these particles that were on the amyloid fibers. She looked at the differential photoluminescence, how bright is it depending on the excitation polarization. And when you see blue, it's more in one channel. And when you see red, it's more in the other. And so interestingly, what Saj found was that if we have a lot of these nanobipyramids, what she could see is that some of them are blue and some are red. There is no significant chirality, and there's definitely no dominant chirality. She then added fibrils to this because we were testing the idea that maybe there's some assembly with the fibrils. And again, some are blue, some are red. The ones that were blue are still blue. The ones that were red are still red. And then finally, she added the THT dye that was in those experiments that we were trying to do that plasmon coupling, and same thing. So, net effect, there's no chirality transfer from the THT molecules to the gold nanorods, and we're not sensing local single particle effects. Saj also looked at this in solution and found roughly the same thing. There's no real effect on the ensemble circular dichrosum from the differential absorption of the small gold nanorod aggregates. And what that tells us overall is that even though we measure an effect in a cuvette, that's an average effect. It could be dominated by a large signal, and it doesn't really correspond to the physics on the nanometer scale. Yeah. Okay, so, one last piece that we're thinking about, and this was Saj's last contribution before she graduated from the lab, was to say, can we use these nanorods to shape light in a more controlled way, in a more intentional way? And so Saj was putting dyes between these nanorod dimers and looking at the emission pattern. And actually, Zek started this project, and Zek was looking at these two nanorods. They could be offset. This is actually a very simple chiral system because the direction of propagation of light breaks the symmetry. And so this isn't just asymmetric, it's actually achiral. And what he sees is that the far field scattering is circularly polarized in the forward direction if we take our laser light and bring it through. He could compute the local fields. He could compute the local electric field as well as the local magnetic field, and using those two quantities, compute the local chirality. And so what happens here is that even though these gold nanoparticles are made in the nanofab and they can't be moved, we can address them with different handednesses of light in order to make a net left circularly polarized or a net right circularly polarized local field. And now we started looking at the emission side. This is back to my microscope that brings in light. It's circularly polarized at the sample, and then the emission comes out. And now in the emission, we're bringing back that polarizing beam displacer. And instead of looking at S versus P, we introduce LCVR number 2. That's basically just a quarter wave plate. And now I can look at right versus left circularly polarized emission. So it's just letting me access a lot of space in polarization. And so again, same experiment. The green circles are dyes that are touching down near those gold nanoparticle dimers. The spots that don't ever fade, those are my lithographic dimers. And what Zek sees is that the detected fluorescence emission will be either right circularly polarized or left circularly polarized, depending on how we address our sample. And so we can use the incident laser light in order to create a single molecule chiral emitter. Finally, Saj asked what happens if we have chiral molecules in this space. And the way that we made a chiral molecule, it's hard, actually, to get a dye that's chiral. And if anyone's thought about dyes, then they're conjugated in a plane. If you twist it, you break the conjugation and they stop being fluorescent. So instead, Saj went back to biology, and she took 2 of those Psi 5.5 dyes that we like. She put them on double-stranded DNA, and now they're coupled very close, and this is a J-dimer geometry. And because they're off by a tiny bit, there's 2 dipoles, and they're a little bit off of each other. It makes a very small chiral center. And so Saj repeated this experiment, putting now J-dimers in that hot spot between the gold particles using those paint experiments. And what she sees is she can detect the dim J-dimers when they're at the center if we use the right polarization. And so at the end of the day, the enhanced J-dimers can be more readily detected, and this is a way of separating out our chiral emission from linear emission. So let me end there. I want to thank the Birnboim family, as well as my gracious host, Humberto, very much for having me here. What I hope I've taken you through is a story of really the last 15 years in my lab, thinking about how dyes and nanoparticles interact. And we're really motivated to think about how we could shape light and apply that to these new biological systems. I think I've acknowledged the people who did the work I was mentioning. I did a throwback picture for Esther. This is my lab. Actually, I could calculate it. This one's 13 now, and this one's 16 now. So, about 11 years ago. And yeah, I'm really happy to take any questions. But thanks again for having me.
Questions?
What are the physical implications of being able to...
Wait.
Hi. So what are the physical implications of being able to measure the chirality of polymers in general?
Yeah. So, I'm going to give you a bigger answer than you asked for.
Okay.
One of the applications would be things like coding or like breaking codes and sensing codes and just having another dimensionality as you're transmitting light. The thing that I'm really excited about, again, based on the fact that we are uniquely positioned to look at these microbial cells, is this idea of, we have a bacteria cell, and it's going to grow out this polymer matrix. And the polymer matrix is chiral. The surface of the cell is amorphous. And so can we find an early detection of when it starts ejecting those fibrils that are pretty small, but they will have a chiral signature and the rest of the cell won't? And that's one example, I think, that would be really interesting for sort of detecting biofilm formation on medical devices or on other infected sites pretty early on. So that's a very specific application. It's the one I'm really excited about. But I think broadly speaking, the more we know, the better.
All right, thank you.
Another question. So I will pass. Thank you.
Hi, I'm curious about the photo bleaching of these dyes, and does the gold accelerate the photo bleaching? So how long could you watch the dynamics of something?
Yeah, thanks. That's a great question. One of the slides I cut last night, because I had too many, was that we actually demonstrated that this enhancement process, it's increasing the total photon budget. So we can make it brighter, but we can also turn down the laser power and make it last for longer. And the reason for this, you know, the big thing that really kills us when we're doing fluorescence imaging is that we keep cycling our molecule up to an excited state, back down, up to excited state, back down. And there's lots of oxygen-related processes that will lead it to photobleach. And so our hypothesis here, which I have not substantiated, is that by increasing the radiative rate, we're leaving the molecule up in the excited state for less time. And as a result, it's less susceptible to these deleterious processes. So overall, the best we did with this very simple geometry was sort of threefold enhancement of brightness and twofold enhancement in length. So about a sixfold enhancement of the photon budget. With engineering, I think you could do a lot better, and this demonstrates that. And that's exactly something I'm really excited about. And then the other thing is if we're having this reshaping, and we could see the molecule, but it seems to be warped, can we live with that as long as my tracks are longer? And there's many circumstances in which they are.
We have time for Jack.
So you mentioned at some point that the nanoparticles shifted the location of the apparent position or the emission, but it wasn't all the way to the center of the nanoparticles. It was a little bit off. So with the two, could you narrow down the position a little bit more? Is that
Yes.
part of your...
Yes, thanks for that question, and I really didn't get to go into the details. So, yeah, in an electromagnetic simulation, we can really predict where the center of that emission pattern should look. The challenge in the experiments is how do I know exactly where that dye was, and how do I know exactly where that particle was? So this is not work that we've done, but others in the field have done really cute experiments using DNA origami. And so you can make DNA origami to control the position of a dye and a nanoparticle really on that nanometer scale. And there you could get that measurement. Now, actually, though, the reason that we got excited about doing these polarization measurements in the first place was that the hypothesis, and this was also from my collaborator, David Masiello, is that, well, okay, you know, in theory, you could do this. So you have these two scattering centers. Where should the center be? If you have two pieces of information, now it becomes a solvable problem. So if I know where the apparent center is, I actually know where the nanoparticle is with high certainty. And now if I know the apparent polarization, that can get me closer there. Now the question is, I still don't have the ground truth of how the dye is oriented. And again, back to other people who are more interested in patterning on a bigger scale. But absolutely, I'm thinking the same way as you.
Trevor.
Thank you for the very exciting talk. So my question is, do magnetic nanoparticles add some interesting degree of freedom? And if so, what would that be?
Oh, it's a good question. I haven't thought about it much. The only place that I've really thought about the magnetic stuff is when we think about chirality. It's the dot product of E and B, and they end up changing. But a magnetic particle, would increase B. It's not necessarily moving it in space. But I bet that would be where it could be interesting to control the chirality in the local field.
Thank you.
Yeah. Food for thought.
One more question, I will be able to run. No questions. So before we end, I want to give Professor Biteen something that we consider special, is a certificate of appreciation that contains a sketch of Birnboim and a sketch of Julie Biteen. Sorry, and these sketches were made by Furkan Çivelek. Where is Furkan? Who's a physics
Thank
student.
you.
So thank you very much.
Oh,
I hope
thank
you like
you.
it.